Hello anatomites, it's Bowles. I know you watched quite a number of videos, but I wanted to do a little summary so that you can practice once you finish your station exploration lab on skin. I know there might be some things that you move through quickly, so I want to emphasize some things that I want to make sure you've got. So first of all, go ahead and use this opportunity to stop the video and go to this Quizlet, either scan the QR code and go ahead and practice some of the structures that you've learned as you're getting ready for your exam. It's important. So do that and then we'll continue. So a couple of things you need to know, and a little saying I have for remembering the layers or strata of the skin. The top of the skin, as if someone were to poke a needle into your skin from top to bottom. The surface at the top. Right after that, it's cows like grass so green, the acronym to help us remember that. A student came up with that a couple years ago. COWS stands for corneum, the top layer, lucidum, granulosum, spinosum, and germinativum, also known as the strata basal. Basal lamina would be below that. Remember, the basal lamina holds the upper epithelial to the lower connective tissue, which is where the dermis is. So don't forget, cows like grass so green, to remember the layers from top to bottom. Looking at those layers, you can see better here. Cornea, cows, lucidum, like, granulosum, grass, spinosum, S here for so, and green is what we call the stratum germinativum, also known as stratum basal. So cows like grass so green. Those are the 5 layers of the epidermis. Now remember, some areas of our body have 5 layers, but most areas of our body are thin skin, and they actually only have 4 layers. So they would be missing what's called stratum lucidum. Going back to this picture, so thin skin is missing this translucent, light can pass through area. So the soles of our feet, the palms of our hands, and the tips of our fingers have lucidum. The rest of our areas don't. Continuing on here are the layers again of the epidermis, and you guys learned this at one of your stations, the different cell types that are found there. I don't know if you can picture or remember what they are. Here's one cell type, here is one cell type, here's another cell type, and we'll continue on here so that you can go over the 4 cell types. So stop yourself and try to remember which is which. Where are keratinocytes, Merkel cells, Langerhans cells, and melanocytes? Try it on your own and then I'll continue and let you know. So the answer, this brown one right here, as you might assume, is melanocyte. So there they are. Notice right here is the dermis, and here the very bottom, stratum basal or granulosum, germinativum, where it grows from. This cell right here, the blue one, is actually a Merkel cell. Notice it's attached to a corpuscle or a sensing cell, which helps us have feeling in our skin. This one right above here is a Langerhans cell. And the Langerhans cells, those guys are kind of like cleanup cells. They act as phagocytes and clean the area. Now up here, all of these cells are called keratinocytes. And those are layers and layers and layers, especially at the top, the dead cells that are sloughing off in the stratum corneum, and they continually die off and are replenished by the growing cells down in the stratum germinativum or basal. Hopefully you got the picture when you learned about the structure of the skin that your fingerprints actually come from the dermal papillae, bumps of the dermis, and connected to the epidermal ridge. That connection holds our skin tightly, increases surface area for connection, and gives us our unique fingerprints, which are unique to each individual. And believe it or not, even in the womb, as a fetus, we begin to have our own unique fingerprints. This is just reinforcing that concept, the idea that those epidermal ridges, followed by those dermal papillae, cause our fingerprints and attach our outer layer to our inner layer in terms of the dermis. And there you go. There's some pictures, and you guys learned during your lab that you have quite a number of sweat pores. They're very, very tiny in between each curve of our fingerprints. I don't know how well you did when you were learning about this, but there are two types of perspiration. There's one type called sensible and one type called insensible. So insensible is, I always think of it as the perspiration that doesn't make sense, and that is the evaporation within your tissues, in those cells, between the cells, and it's just normal while you're sitting. Not sweating and exercising, just sitting. Sensible is where you're excreting water by sweat glands. And remember, there's two types. There's the kind that smells and the kind that doesn't. I don't know if you guys remember this animation, which talked about this process. Here it is.
In the endoplasmic reticulum, they are processed by the Golgi and then released through exocytosis. Most glands in the human body are merocrine glands, such as the salivary glands, the glands of the pancreas, and the eccrine sweat glands, which release the watery sweat for thermoregulation. Apocrine glands release a portion of the cytoplasm which is pinched off from the apical portion of the cell. There are glands known as apocrine glands in humans, which produce the type of sweat which results in body odor, and mammary glands were considered to be modified versions of this type of sweat gland. Recent research has called into question whether true apocrine sweat glands exist in humans, and whether the glands formerly known as apocrine sweat glands in humans should better be classified as merocrine glands. So this will require further study. In glands classified as holocrine glands, dividing cells synthesize a number of products, and then the entire cell ruptures, and it is the contents of the cytoplasm then which constitute the secretion of this gland.
So remember those types? Different kinds of perspiration. The kind that smell are the eccrine kind. The kind that don't, they just produce watery secretions, are the merocrine kind. Couple more things. Skin color. Depends on the pigment melanin, but also the pigment keratin, which is actually a yellowish or orange pigment, and circulation, or the amount of red blood cells or blood vessels that come to the surface. So our skin color is actually determined by those three things. And just keeping in mind, the melanocytes are at that lowest layer, the stratum basal or germinativum. And notice here that they send out into the upper layers their pigment, especially when we're exposed to sun, in a form of melanosomes, which is kind of like a little vesicle that comes out from the melanocyte, and it goes into the neighboring keratinocytes, those cells, and that's why suntan will go away as the layers grow out. Okay? Just on the topic of melanocytes for a second, you've probably heard of albinism, albinos. Albinos are people or organisms that are missing pigment. They're not missing melanocytes. They're actually missing melanin, the pigment that causes us to have our skin color. So you notice their characteristic lack of color. There's a couple other things related to coloration. Sometimes skin has a bluish tint. That's because of lack of blood flow. So remember, blood flow does contribute to our tone or skin color. Sometimes you can visibly see that around the lips and in the fingers. In fact, it's checked for to make sure people are properly oxygenated in the medical profession. You might also see things like jaundice, or in the case of Addison's disease, a darkening of the skin related to various conditions. Vitiligo, maybe you've heard of that before too. That's actually when people are losing color, and that is due to a loss of melanocytes that causes that. And then I also want you to go ahead and do this. So stop and take some time to go through this matching to allow you to practice again and review. And then there's some other reviews I also want you to do, and go ahead and take some time to go over these as you study for your quest, a short test on the skin. So So make sure you know the layers, and you can name structures in each layer. What are the 5 layers of epidermis? Don't forget, cows like grass so green. The cell types, and here's a little matching to help you with the cell types. And then there's a few more. What are the answers to each of these questions? Basically, to help you prepare for your exam. So be prepared to answer these. Stop now and write some notes, practice, and get ready for your quiz. Good luck, anatomites.